Welcome to this explainer. Today we are embarking on a real biological detective story. Picture this. You're a researcher. You've just wrapped up a massive sequencing experiment, and now, well, you're staring at an absolute mountain of genes. How do you trace those biological clues back to their precise origin in the body? Today we're going to explore a really powerful computational magnifying glass designed to do exactly that. It's called TissueEnrich, and we're going to break down exactly how it works, how it processes all that data, and how you can actually use it to uncover hidden signatures in your own research.
So here is the core dilemma researchers face every single day. You run an RNA-Seq experiment, maybe on a mysterious cluster of cells or perhaps a newly discovered tumor phenotype, and your output is just this massive pile of clues. Basically, a long list of highly expressed input genes. But a list of genes alone doesn't really tell you the whole story, right? What you really need is to pinpoint the biological culprit. You need to know what specific tissue are these genes actually pointing to. Are they brain-specific, liver-specific? That's the exact mystery we need to solve, and trying to do it manually across thousands of genes is practically impossible.
Okay, let's dive into this. Here is our roadmap for today. 1. Meet TissueEnrich. 2. The data engine. 3. Defining tissue specificity. 4. The math behind enrichment. 5. Running your analysis. And finally, 6. Interpreting your results. Let's start with Section 1, Meet TissueEnrich. Authored by researchers Ashish Jain and Gitu Tuteja, TissueEnrich is available as both a bioconductor R package and a super accessible web tool. Its entire purpose is to solve the exact dilemma we just talked about. It is specifically designed to calculate the enrichment of tissue-specific genes within a given set of input genes. So, for instance, if you input a list of the most highly expressed genes from your RNA-Seq data, or even a set of gene co-expression modules, TissueEnrich steps in as your biological detective. It determines exactly which tissue-specific genes are statistically enriched in your dataset.
Moving on to Section 2, the data engine.
To give TissueEnrich its analytical superpowers, the authors tapped into some of the most robust RNA-Seq databases in the world, processing data from 3 massive repositories. First up, the Human Protein Atlas, which covers 35 human tissues. Second is GTEx, providing data across 29 human tissues. And third, the Mouse ENCODE project, covering 17 mouse tissues. Now, what's really interesting here is the quality control behind all this. To make sure the calculations are completely solid, the tool specifically relies only on tissues that have at least 2 biological replicates. Oh, and a quick pro tip, if you ever see a tissue prefixed with an E, like E14.5-placenta, that simply denotes an embryonic tissue from a specific time point. All the others without that E are isolated from adults.
All right, Section 3, defining tissue specificity. But how do we actually define what makes a gene tissue specific in the first place?
Well, the Human Protein Atlas algorithm uses a really strict classification matrix to categorize every single gene. This is based on expression levels, which are usually measured in transcripts per million, or TPM, and fold changes. If a gene has an expression level less than 1 TPM across the board, it's simply categorized as not expressed. But look at the strict criteria for actual enrichment. A gene is considered tissue-enriched if it has an expression of at least 1 TPM, and its expression is at least 5-fold higher in one particular tissue compared to all the others. If it shows that same 5-fold spike, but across a small group of 2 to 7 tissues, it's called group-enriched. And if it's 5-fold higher in one tissue compared to the average of all others, we call that tissue-enhanced. Anything else falls into expressed in all or just a catch-all mixed category.
So the absolutely crucial takeaway here is this. Out of those 6 categories we just looked at, only 3 of them are actually used for the enrichment calculations. When TissueEnrich talks about tissue-specific genes, it is exclusively referring to genes that fall into the tissue-enriched, group-enriched, or tissue-enhanced groups. These are the genes with a strong mathematically proven preference for certain tissues, which makes them the perfect biological markers for our detective work.
Section 4, the math behind enrichment. Let's look at how we prove these clues aren't just random chance.
TissueEnrich relies on a real statistical workhorse known as the hypergeometric test. It essentially calculates the probability that your input genes overlap with the tissue-specific genes purely by chance. But you actually have 2 distinct ways to run this engine. If you don't provide a background, the tool defaults to comparing your input against all the genes in the chosen dataset. However, if your experiment only looked at a specific subset of genes, you absolutely want to upload that subset as your background gene set. This ensures the math accurately reflects your specific experimental conditions, rather than comparing against the entire genome.
Just remember, your background set has to contain all the genes present in your input set.
Now, because we are testing across many different tissues simultaneously, we naturally run the risk of false positives. But TissueEnrich handles this mathematically. The raw p-values from that hypergeometric test don't just get handed straight to you. They are rigorously corrected from multiple hypothesis testing using the Benjamini and Hochberg correction. This means the final numbers you are looking at are highly dependable and mathematically controlled for false discoveries.
Section 5. Running your analysis. So, how do you actually run this thing?
The web interface is wonderfully seamless and user-friendly. Step 1, you simply go to the Tissue Enrichment tab and either paste your list of genes right into the text area or upload a text or CSV file. Just make sure there is one gene ID per line. Step 2, you select your options, your organism, your gene ID type, and your dataset. And what's really fascinating here is that you can even analyze cross-species data. You can input a mouse gene list, select mus musculus, but then choose a human dataset like the Human Protein Atlas. TissueEnrich will automatically use one-to-one orthologs between human and mouse downloaded from the Ensemble version 91 database to run the comparison. It's incredibly clever. Finally, step 3, just click Submit.
The moment you hit Submit, you are rewarded with a highly dynamic set of visual data. It doesn't just hand you a static spreadsheet. You get an interactive bar chart right out of the gate. If you hover your mouse over any bar, you'll immediately see the tissue name, how many of your input genes are specific to that tissue, the fold change, and the statistical enrichment score, which is plotted as the negative log 10 of the adjusted p-value. Want to dig a little deeper? Just click directly on one of those bars. The tool instantly generates a heat map, allowing you to visualize the specific expression details of those genes in that particular tissue. You even have a histogram toggle if you'd prefer to visually plot the bars by fold change instead of p-value.
And finally, Section 6, Interpreting your results.
Point 0.01. According to the creators of TissueEnrich, an adjusted p-value of 0.01 is the strictly recommended threshold you should use. If the adjusted p-value for a tissue falls below 0.01, you can confidently declare that your gene list is enriched for that specific tissue. If it's above 0.01, well, the evidence just isn't strong enough to make that claim.
And here is a crucial best practice straight from the tool's documentation. Always check if the tissue you care about is actually in the dataset you selected. For example, imagine you are studying a list of trophectoderm-specific genes from a single-cell RNA-Seq analysis. Because the placenta is derived from the trophectoderm, you would strongly expect to see placenta tissue enrichment. But if you chose the GTEx dataset for your analysis, you would find absolutely nothing. Why? Because the placenta simply does not exist in the GTEx dataset. It is entirely absent. However, it is present in the Human Protein Atlas dataset. Your choice of dataset entirely dictates your discoveries, so always know what's in the library before you start searching for books.
Because of dataset variations exactly like that placenta example, the ultimate best practice is cross-referencing. Don't just rely on one database. If the target tissues you're investigating happen to be present in the Human Protein Atlas and GTEx and the mouse ENCODE datasets, you should run your gene list against all three. True confidence in your biological detective work comes when you see consistent enrichment results across multiple independent datasets.
At the end of the day, TissueEnrich takes the overwhelming complexity of big data and turns it into a clear, visually interactive narrative. It gives you the power to trace a seemingly random pile of genomic clues directly back to their biological source. So, as you look ahead to your next experiment, ask yourself, which hidden tissue signatures are waiting to be uncovered in your next RNA-Seq dataset? The tools to find them are right at your fingertips. Thank you so much for joining me for this explainer, and happy analyzing.